For our next process, we're going to talk about how a protein is made. And so what we have to do is we have to take our DNA, we have to read its code, and the code of DNA is going to give us the information to build a protein. All proteins come from the DNA code. And so this is a two-step process called transcription and translation. So we're going to go over the first step called translation in this video, and we're going to look at Handout 2M. And in Handout 2M, we're going to discuss this first step in making a protein from our DNA. This step is called transcription. And what transcription is turning our DNA into RNA. This seems kind of weird, but despite how important DNA is, our cells actually cannot read DNA. So we cannot make a protein directly out of DNA. We have to turn DNA into RNA first. Now, this is done very similarly to what we talked about when we talked about DNA replication in the last video. So what we have to do is we have to unzip our DNA, just like we did before with helicase, and then we have to read the DNA, except this time, when we read the DNA, we're not going to make more DNA, we are going to make RNA. And so we have a different enzyme called RNA polymerase that's going to jump onto the DNA. It's going to read our DNA code, but it's going to make a string of RNA that carries the DNA message. So we call it messenger RNA, mRNA, because it's carrying the DNA's message. All right, now what we can do after we have this messenger RNA is we can use the messenger RNA to make the protein, because the cell can read messenger RNA, but it can't read the DNA, even though DNA is so important. All right, seems weird. So what's needed? Well, we need our DNA to be read. We need that RNA polymerase to read it. And then we need RNA nucleotides instead of DNA nucleotides to get stuck in the correct sequence, forming the messenger RNA. So this is pretty simple. It says what's created. Well, what we create is this messenger RNA that then can be read by a cell. And that's all there is to it. This is pretty simple. And that's really all you need to know about transcription. So now we've got this messenger RNA. And once we have the messenger RNA, we can use this messenger RNA to build a protein. And that's going to be the next video, a process called translation, which is the second step in making a protein. All right.